I think I should start with definition, because by the biobank and biobanking, the definition has been quite controversial over the last 18 years, since the term first appeared officially in 2001, and it was introduced by the OECD in 2001, the Organization for Economic Cooperation and Development. So now, after these 18 years, there is an ISO norm, specifically which focuses on the requirements for biobanks. This is the ISO 2387. So the ISO definition of biobank and biobanking is the following. Biobank is a legal entity or part of a legal entity that performs biobanking. And biobanking is the process of acquisition and storing, together with some or all of the activities: collection, preparation, preservation, testing, analyzing, and distributing biological material and associated data. In other words, a biobank does not have necessarily to be a laboratory. A biobank may be just receive and store specimens is a biobank. A biobank can have a laboratory, not performing any analysis, so receiving samples, processing, preparing the specimens, and a biobank can have also an analytical laboratory performing some analysis. Also, a biobank may be embedded in a clinical biology laboratory, but also or a pathology laboratory, but also may not be embedded in a clinical pathology laboratory. And as we know, the clients are different. So the clients of a clinical biology laboratory, your clients are clinicians. However, the clients of biobanks belong to a different tribe. The clients of biobanks are clinician researchers, university researchers, industry researchers. And talking about research, you may have seen these big titles in big journals over the last 5 years about the irreproducibility of research results. So, irreproducibility of research results, why is this? This is a research laboratory. It's not a clinical biology laboratory, to be clear and avoid misunderstanding, but these are things happening in a research laboratory. For example, you can see expired consumables, missed maintenance of equipment, experiments being performed under different conditions, suboptimal data tracking, also data security with post-its floating around and shared passwords, messy storage in the refrigerators, the freezers, and so on. It's a research laboratory. A lot of what has been said here has focused on the human cost and the financial cost linked to the situation where preanalytics go wrong in a clinical laboratory. However, there is also a big financial cost associated to when preanalytics go wrong in a research laboratory. This is an economic analysis recently performed, which shows that in the United States only, the annual cost due to irreproducible research results is of the order of 28 billion euro in the US only per year. And a big part of this cost incurred to irreproducible research is linked to biological samples, reagents, or lack of reference materials, and so on. So a big part of the problem is linked to what happens before even the samples reach the researcher's laboratory and the bench. So things like, for example, inclusion criteria in a research project. We have seen publications with cancer subjects being male and female subjects being female. We have seen things happening during sample collection. Again, we have seen publications where cancer specimens have been collected in one clinic and the control specimens in another clinic. Problems linked also to specimen processing and storage, and we have seen there publications where cancer specimens have been stored for 10 years, but control specimens only for 1 year. So all these things here linked to collection, processing, storage of samples, this is what biobanks do. This is the core business, this is the focus, and this has to do with what we call a processing method. So I would like to focus on this concept of processing method, because I know you are not used to hear this term and this concept, but what is in the red box is what is the core business of a biobank. The biobanks are not there to produce measurements, analytical results. They are there to produce specimens. So what is in the box is our definition of a processing method where the input is a specimen, for example, a whole blood, and the output is another sample, for example, the extracted DNA or the extracted CF DNA. And this is the product. This product then will certainly go into an analytical process to produce, at the end, some measurements. But, as I said, the core business of the biobank is this processing method. Now, what are the quality management system components that are linked to a processing laboratory such as this of a biobank? First of all, what has to do with consistency and competence, and here, as I said, we have this new ISO 2387 published last August. This is an accreditation standard. It's at the same level as the 17025, at the same level as the 17034, requirements for reference material producers, and this is the requirements for biobanks. A second element is linked to the, of course, critical pre-analytical steps. Here, I'm sure you're familiar with these technical specifications published by CEN Technical Committee 140, where in the scope of these technical specifications are not only the clinical laboratories, but also the biobanks. So this has to do with critical pre-analytical variables for each process, RNA extraction from blood, from frozen tissue, from FFPE tissue, and so on. And this is a continuous work where the SPEED-FOP consortium is being very active. Of course, these technical specifications focus primarily on clinical biology laboratories. They may be a bit restrictive sometimes for biobanks because our field of action is larger, but still they're very useful. A third element has to do with the traceability of pre-analytics, and here yesterday already you heard about the SPREC. This is the sample standard pre-analytical code. This is something that has been published already 9 years ago. Now we are at version 3, and this is a simple 7-element or a letter code which summarizes in 7 letters the most critical pre-analytical variables for each type of specimen, fluid or tissue. For the fluid samples, these 7 elements are the sample type, the primary container type, the time and temperature before centrifugation, the conditions of the centrifugation, 1 or 2 centrifugation in terms of speed, break temperature, the time between centrifugation and storage, and the temperature, and finally the long-term storage conditions in the container. So 7 letters to summarize the pre-analytical history of our samples. A fourth element related to quality management in biobanks has to do with the methods, so these processing methods. Of course, where there are manufacturer recommendations, as everybody else, we have to follow them. However, if there are not, or if we change something, or if it's a homemade method, we have to validate the method. And in this now specific context, I would like to mention also this ISO, which is not yet published, so this is at the DIS stage currently, ISO 21899, which focuses specifically on the verification of a processing method in biobanks. So a little bit more about that. Validation of a processing method. It's not about, it's not an analytical method, so it doesn't have to do anything with quality control materials, reference materials that we use in our analytical method. But it does have to do with in-process quality control materials, like the one which was previously mentioned, which is a spike-in reference material that we use in the process to monitor the performance of an extraction method. Processing method doesn't have anything to do with logistics. For example, labeling of the tube, so anything which does not influence the intrinsic properties of the sample that is in the tube is not a processing method, but anything that influences the intrinsic properties of the sample which is inside the tube, which influences the yield, the integrity, the purity. This has to do with the processing method. Validation of a processing method is not about validation of the materials, of the samples. In other words, something that has to be validated on each and every aliquot of the material produced, like the authenticity or the representativeness of the material. This is not validating the processing method. Validating the processing method has to do with everything that, for example, validating the output in terms of yield, in terms of integrity, so that once we have validated our method, we are confident that all the samples that will be produced with this method will have the same approximately quality attributes. And finally, we are not in the therapeutic area, so ICH guidelines are not in the scope. However, REMCO, ISO REMCO reference material producer guidelines might be helpful in this area. How do we validate a processing method? These are the main things that we will look at. We will look at fitness for purpose. In other words, is the output sample suitable, fit for purpose for downstream analysis by mass spectrometry, by genotyping of the cfDNA, and so on. Fitness for purpose. Reproducibility, when we put in our DNA extraction the same specimen of the novel, do we get as an output a sample that has the same quality attributes? Robustness, and this is robustness to preanalytics, so the robustness of the method to extended times or temperature or centrifugation conditions. These are things that we have to validate. The homogeneity, in case we produce many aliquots of the same sample as an output, we would like to be sure that all the aliquots are homogeneous, and the stability, which is the long-term stability of the output samples. In order to validate all these things, of course, we have to perform some analysis, because we are going to study either the concentration or the purity or the integrity, the cell viability, the cell functionality. Depending on the method, we will choose what is the most relevant analysis to do this validation. Moreover, there are some more elements in the quality management system which are of interest. As I mentioned already, the use of in-process quality control material. This is especially relevant when the processing method is a challenging one, or when the output may be very low in concentration. For example, when we extract DNA or RNA from FFPE material, where if the tissue is totally necrotic, we may get nothing, but still we want to validate that the method has properly worked. So the use of in-process quality control materials is useful. We also use what we call range charts. So this is all the samples that go through for example, cfDNA extraction or DNA extraction from blood. We monitor the characteristics in terms of yield so that we can see if there is a trend, if suddenly the extraction method, something goes wrong. This is the range charts. And another element is the participation in EQA processing schemes. So this is again something I think you're not familiar with, because you know very well the EQA analytical schemes, but we also have EQA processing schemes. The concept here being that the participants receive, for example, a stabilized whole blood. They extract the DNA, and they send all the DNA to the central laboratory, which performs the analysis for the yield, the purity, the integrity, and they receive the report to see how their DNA extraction method performs in terms of yield, integrity of the output DNA. Okay. There are challenges in the validation in this quality management system and the validation of processing method. Challenges may be linked to the fact that when biobanks are embedded in clinical biology laboratories. Why? Because obviously the focus of clinical biology laboratory is, very rightly, the clinical diagnostic assay. It's not so much the processing method. Another challenge linked to exploratory methods, and by exploratory methods we call any methods that may be in-house produced or modified standard methods, or a method that is new, that has never been performed before. Such exploratory methods obviously may be difficult to validate. Other challenges here are linked to the validation of these analytical methods, because these analytical methods that we have to apply to validate our processing methods obviously are not clinical diagnostic methods. DNA quantification, RNA integrity, these are not clinical diagnostic methods. So there can be a challenge. The interdependency between this continuum container, processing, and analysis. Because when we validate a processing method, this is for the workflow, container, processing, in the scope of a specific analysis. If the container changes, if it is to validate in the scope of a different analysis, we have to start all over again. There are special challenges linked to the use of in-process quality control materials and the EQA materials, because in terms of homogeneity, in terms of stability, in terms of volume, if we want to use CSF, we cannot have big volumes of CSF easily, so sometimes surrogate materials may be used. So the production of these materials also is a challenge. A second now issue area which has to do with the quality management system in biobanks and processing laboratories is the one related to the qualification of samples. This is a technical report published some years ago by ISPER is the International Society for Biological Environmental Repositories. There is a working group on biospecimen science where we published this report, which is a summary of the assays that are currently available for the qualification or quality stratification, I will go into these terms in a second, of the biospecimens. So the definition, what we call qualification of a specimen, is the verification of the specimen's fitness for purpose for use in a downstream research area, which can be a disease area or a type of analysis like the mass spectrometry. A sample may qualify or not qualify for such downstream use. It is a yes or no. The second concept is the concept of quality stratification. So this is a categorization of specimens. We put the specimens in category according to their quality characteristics, so that, and these characteristics may be linked to technical preanalytical factors or biological preanalytical factors, but the purpose here is to put them in such quality categories so that these characteristics can be used as statistically as covariables by the researchers when they're going to use these specimens for their research. So qualification and quality stratification. Clinical diagnostic results are very often, and they should be, part of the characterization of the specimens, because researchers also need very often clinical diagnostic results to have a full characterization of their samples. So quality control of samples. Now I would like to go a little bit deeper and highlight these concepts of characterization with few examples. So one example on each slide. When we say assays for qualification of specimens, here the concept is that in order for a specimen to qualify for use in a specific disease area, we would look at biomarkers which are known to be of relevance, clinical relevance in this disease area, and which are also known to be sensitive, labile to preanalytics. The idea being that if these markers are below the detection limit of the method that we use to measure them, the sample is not fit for purpose, for research, for discovery of new biomarkers in this disease area. Because if these biomarkers cannot be detected, we're not talking about any threshold, we are talking being below the detection limit of the method. It means something has gone wrong with this sample, it is not fit for purpose for research in this disease area. The second concept of quality stratification or categorization in quality categories. Here also now let's see an example. If we deal with serum, we can put the serum samples in categories, in quality categories, according to different relevant factors. It can be effective coagulation, so by measuring fibrinogen below a certain level to assess that the serum has been effectively coagulated. Hemoglobin contamination, at least to put the samples in 2 or 3 buckets, quality categories, so that the users can know and make their own assessment if they want to use the sample or not. We have similar concepts in the area of tissue, fresh frozen or FFPE tissue, so these are just examples. And here we can also apply some quality categories to highlight the fitness for purpose of tissue specimens, formalin fixed paraffin embedded, for example, DNA, for use in downstream NGS, CGHRA technologies, based on specific thresholds. So here, for example, we can apply a delta CT, the Illumina FFPE QC assay, based on whether or not the delta CT higher or lower than 2. The specimen can be put in 2 categories, which qualifies for NGS or not. Similarly, by amplifying different sizes of amplicons, if the amplicon higher than 200 base pairs, the DNA can qualify for use in CGHRAs, and so on. And similar examples again also exist for PBMC, for cytological specimens, where we can put viable PBMCs in 2 or 3 quality buckets, categories, which also are related to the time between blood collection, venipuncture, and isolation, fecal isolation of the PBMCs. How? By measuring, for example, contamination by CD15 positive granulocyte, because this is an indication of long precentrifugation delays. For the stool, we can assess the presence of PCR inhibitors by a SPUD assay. These kind of quality markers for analysis are applied during the initial validation of the processing method, but also during a continual or the monitoring of the performance of the processing method. And here I will show 2 examples. Even for easy methods, even for easy, not complex, not challenging methods, like so if we have a DNA extraction from blood, there is the initial validation, but also, for example, once a year, over a random percentage of samples, we like to see the performance of this extraction, DNA extraction method from blood, by looking at the yield. So where we know that we expect to have above a certain yield. We expect to have above a certain percentage of double-stranded DNA, because this is an important quality attribute for sequencing. We expect to have a DNA integrity above a certain level. We expect to have amplifiability above a certain level, and we expect to have absence of PCR inhibitors. And again, in another area where, again, this is a very simple method, this is the method which is plasma aliquoting from EDTA blood. It's just about aliquoting, but also many things can go wrong in this preanalytical area of simple plasma aliquoting. So again, as part of the monitoring of the processing method, we would look at quantity of platelets in the plasma, where we expect to have below a certain level. We would look at presence of hemoglobin, so this you know very well. And this is an assay which is not well known. We call this the LACA score. We published a few years ago. This is the ratio of ascorbic to lactic acid, because this changes significantly with the precentrifugation time. Okay, so enough about this. And going to the end, there is the component which consists in quality management system in EQA schemes. So I mentioned to you the processing EQA schemes, which have to do with the processing methods. There is also, so we in IBBL we decided to provide this program because it did not exist, so that people could benchmark and compare the performance of the analytical methods that they have to perform for the validation of the processing method. So the DNA quantification, the DNA integrity measurement, so CD40 ligand, there is not anybody who performed this measurement, but cell viability, tissue histology, percentage of tumor cells. This is not a clinical assay, but this is something, this kind of PT program is very useful for biobanks. And the last tool also that we made publicly available, and we can take a look, is what we call the findmyassay.com. So this is an online tool where you can go, you can put your specimen type, you can put what you want to use it for, and the tool will show you what would be the most relevant quality control assays or qualification assays that you can perform to better characterize your sample. Okay. So as a conclusion, there are many things applying to the quality management system, the preanalytics in biobanks and processing laboratories. We highlighted 3 of them. The first one has to do with the traceability of the preanalytics. There we mentioned the SPREC, the sample preanalytical code. A second point has to do with the initial validation and the continual monitoring of the performance of our processing methods. And a third element which has to do with the characterization and quality stratification of samples. Clearly, such qualification, these quality control assays, we do not perform them all the time on all the types of samples that we produce, for sure, depending on the type of collection, depending on the type of sample. Some of these qualification assays are performed on all the samples that are produced. Some of them are performed on all the samples that are distributed upon distribution. Some of them are performed on a special demand by a client or when there has been a non-conformity, and some of them are performed once a year on a random selection of samples. Okay, so to finish, I have 5 questions. The questions may seem very similar, but they are not identical. There's a small difference in the wording. Okay. So the first question is, and this is a poll to see your opinions. For clinical laboratories, are preanalytical processes, the pre-examination phase, okay, very good, of clinical diagnostic assays, so clearly, the pre-examination phase of research assays, or processing methods, that produce specimens that are not necessarily belong to a total analytical workflow. Okay, so I expected the first, of course, we are in the context of a clinical laboratory. Good, so we can go to the second question. Is this the second? Yes. Okay, the second question is the same. So what are pre-analytical processes, but for biobanks embedded in clinical laboratories? Okay, so there is a shift. Okay, I'm glad to see that you can see the difference of the focus. So biobanks embedded in clinical laboratories, there we have the pre-analytics, which are the pre-examination phase also for research assays, and also part of, not necessarily part of a total analytical workflow. And the next question, very similar, but this is now for biobanks which are not embedded in clinical laboratories and which serve a wide variety of end users. Very interesting. Okay, thank you. So we can see that it's kind of like a continuum, but we have a different focus and also different, probably, definition of the pre-analytical workflow. Thank you. I would like to keep these statistics, very interesting. And the last two questions, again, these two questions look similar but are not identical. So in clinical laboratories, are in-process QC materials and EQA processing schemes relevant in the context of specific clinical diagnostic assay? Relevant independently of any specific clinical or research assay, so there we have the majority. Relevant only for complex, challenging, exploratory, or homemade methods? Irrelevant. Oh, someone said irrelevant. Okay, so the majority independently of specific clinical or research assay. Okay, thank you. Which goes into towards the sense or the concept of a processing method. But also, I think they are relevant. Okay, we have a lot of response. For me, such in-process QC materials and EQA processing schemes, they are also relevant in the scope of specific clinical diagnostic assays, when, especially for like the cfDNA and so on. And the last question is the same, but this is not in clinical. This is in biobank laboratories. So in-process QC materials and EQA. Okay, and then again, we can see the shift. So these are relevant independently of specific assay, and much less relevant in the scope of a clinical diagnostic assay. So thank you very much. And with this, finished.